We need to get our microphones on for the actual feed for the YouTube. Ivan, if you could put that over your other ear, just so there's no feedback between the two microphones. Exactly, yeah. And then, let me just see here. There we go. Does that work?
I guess.
It sounds like it works to me. Hello, everybody. Does that work?
Yes. It's better.
Okay, good. Nikolai, does that work?
Yes.
It's on the YouTube, is it? Excellent. It was, first of all, Ivan, we met in a trampoline park.
Yep.
It was a few years ago. There was a concert in a trampoline park, and we got chatting about just brainstorming just ideas to make cool things happen, you know. And then I was calling you up a couple of months ago and asking advice about how to bring this concept, science and beers, to a festival. And then you started telling me the story of what you were doing at the moment. And I just found it so unbelievable that I wanted you to stop telling me the story and wait until I got a recorder. And so we could make it into a live podcast. So I've on the phone, just in passing, he starts telling me these stories about Nobel Prize winners from 100 years ago, about methods used in the Second World War to cure venereal disease.
You're like, who is this guy?
About an AI to track COVID-19. I thought, okay, this is too much. This is too much. Let's sit down. Let's get a beer and talk about this thoroughly. So, first of all, a bit about yourself, Ivan.
What do you want to know?
So, also this guy's just crazy busy. You know, you came from Norway to study
Medicine.
an MD, to be a medical doctor. While you were doing that, you got an actual academic Ph.D. Yeah?
Yeah. At the same time.
At the same time. And you started a company.
Yeah.
Tell me about the Ph.D.
The PhD?
Yeah.
Well, it was, I had studied medicine for 3 years, and I was curious about science, because I heard it was hard, and you had to have stamina, and you had to be creative. So I thought, that sounds really fun. And then, so I took a break in my medicine study, and I did a science year. And during that science year, I started developing a method. So when my master started, the last 3 years, I continued developing that method, and it turned out that I could make my own PhD protocol. So I made my own PhD protocol, and I almost forced my main advisor to be my main advisor, because nobody wanted to be my main advisor, because I was just, you know, going on my own path, sort of.
So you're in the middle of studying medicine, and you didn't think that was scientific enough.
You want the honest answer or the diplomatic answer?
It sounds that way to me. Of course, the honest answer.
Yeah. Well, of course. Yeah, I wanted more science, for sure. And I was looking in the medicine books, and I was, when you look at the brain, you have all these neurons, but it does not say very much about how they connect. So it's like looking at a web that is made by spiders, but no one actually tells you how the web works. It only tells you how separate parts work. So I was starting to get curious about, does that brain actually work as a collective unit in this neural network, or is it correct that it doesn't exist? Because that's the truth today. The truth today in the medicine books that we all read is that the brain works in silos. It doesn't work. It doesn't work as one collectively biological mass.
Well, yeah, you see these images of the brain, and you have it divided into sections. You have the hippocampus,
yeah,
for example.
Exactly. So, for example, if you have you have hippocampus, you have amygdala. You know that if you have a lot of anxiety, it is associated with a more activity in amygdala and these things. But how the whole brain works as one organ, it doesn't say very much about, and especially also the cerebellum, the little brain. You have cerebrum, which is the higher brain, and then you have cerebellum, which is the little brain. So, for example, in dementia and very many brain diseases, it does not talk about the little brain. It's like it doesn't exist. Of course, in science it does. It's done a lot of science on it, but when it comes to treatment in the hospitals, it doesn't say very much about it. So this was a sparkle of curiosity. And then I went to science dating.
So tell me about the curiosity there. You're thinking, okay, is nobody writing about the brain as a whole?
Yeah.
Is that true? Nobody is trying to figure out.
No, it's not true.
Okay.
People are trying to figure out.
Yeah.
But in the medicine books, what is the truth? What is written in them is often lagging in maybe 10 or 20 years between what we are producing of science today.
Mm-hmm.
And of course, that's the one reason why when I was reading in it, it didn't say anything about how the whole brain works as a biological mass. But the other reason is also that even though there are many people doing science on it, in order to actually make it a scientific truth, it's not enough that you produce a few studies. You have to produce a big amount of studies. Mm-hmm. And that we have not done so much of today.
Okay.
So in short, it doesn't say much about the brain as a whole organ.
So then you were inspired to try to do your bit to contribute to the description of the brain as a whole.
Well, I was just curious.
Yes.
Because I never thought I would be do brain science.
Yeah.
I played football when I was young, and I went to the military, and I always enjoyed doing physical things, and I thought science was for old people with glasses that didn't have any other hobbies to do. So I was just curious. That's the end of story. Yeah. I have no expectations, and I had no goal of making a Ph.D. or,
It's just pure curiosity driven.
Pure curiosity.
Okay, so what happened next?
Well, then I went to science dating, where you meet all the doctors from the hospital, different departments. Like
speed dating with doctors?
So of course, I went to...
Has anybody been on a speed dating with doctors before? No?
Or scientists.
Scientists, yeah.
Yeah. So, and then I looked at the program, and then I saw it said Department of Nuclear Medicine. And then sort of like I forgot my interest of the brain. I was thinking, nuclear medicine? What is that?
That sounds cool.
So then my curiosity actually made me go there first. And there were some beautiful people there. And then they told me about nuclear medicine. And it was even more fascinating than what I thought it was. So then I learned about that.
Do you mean nuclear medicine? Are you talking about the treatment of cancer, or?
I'm talking about nuclear medicine.
What?
Nuclear.
Like what?
What do you associate nuclear with?
Bombs.
Yeah, which comes from radioactivity, right? So it's medicine made out of radioactivity. So at Odense University Hospital, they have 2 cyclotrons which produce radioactivity every morning. And then you inject it into the blood veins of the patient, the radioactivity, and then you can label it with whatever chemical you want, sugar or protein, and then you take an image of the patient. So you can actually see how the biology is based on that you inject radioactivity. So that's nuclear medicine, in short. Yeah.
Okay. So you were interested in that, and then you came back to the brain.
Well, the thing was that I went to the Department of Neurology, Neurosurgery, but they didn't have a project that was exciting. Because, if you want to look at the whole brain, you cannot look in small Petri dishes. That's impossible, because there you have maybe brain cells. If you look at mice, well, you're looking at the brain of a mouse or a rat. And I wanted to look at the human and the whole brain. So in that sense, it made sense to actually study nuclear medicine, because it imaged the whole brain. It's a biological footprint of the brain. So, yeah.
And was this for the masters, or was this the beginning of the PhD?
No, this was just the beginning of my research, I don't know, curiosity.
Yeah.
So then I went to the Department of Nuclear Medicine and I said, I want to study the brain in your department. And then they said, we have no studies about the brain. And I said, no, I don't believe that. And then I went to 2 or 3 meetings, and then finally they said, well, maybe there's a doctor who has some images in in his office. And then they had 50 images of brain tumor patients. So after 3 meetings, they said, okay, you're allowed to do science here. And then they had 10 other students, so they didn't have time. Nobody wanted to be my main supervisor, but that actually fitted me well, because I really was so curious that I wanted to do my own exploration, and of course finally I got the main supervisor, which was the head of science at the department, and I learned very much from him.
And did you set out with the research question?
Yeah.
Which was?
Well, you mean for a thesis or just in general?
Well, in general.
Well, it was, of course, I had to read up a little bit, but it was, how does diaschisis affect patients with brain tumor?
Yeah. So I know that the discovery is to do with that. So I think we need an introduction to diaschisis.
Yeah.
Diaschisis. Can you give us an introduction to that?
Yeah. Well, it means it's a Greek name.
Yeah.
And in English it means shock throughout. And it was discovered in 1914 by a very famous neuropathologist, von Monaco. But today, if you ask someone at the, well, this is not him.
Okay.
This is 3 other guys.
3 other guys. Okay. Forget about that. Forget about that.
But they are related, of course, to the hypothesis. But this actually, So diaschisis means that he investigated and he found that if you have a disorder in your brain in one location, that will actually do that the neuron will also be damaged in a different location. That's diaschisis, shock throughout. It's just like a shock throughout the whole neuron. So it's like if you call me from Ireland and I'm here in Denmark and you have an old-fashioned phone, then if you get the damage in Ireland, then of course you will lose the signal in Ireland, right? But I will also lose the signal.
Yes.
I cannot hear what you say if it was an old-fashioned phone. But that's diaschisis. It just means that the whole neuron is damaged.
So, if he was able to figure that out, then that was evidence that, the connection between different parts of the brain is, there is a connection between different parts of the brain, So if I get a hit in the head over here on the right side of my head, it will not only damage the cells close to the impact, it will damage the cells on the left side of my brain.
Yes, in theory.
In theory.
Yeah, that was the theory of diaschisis.
Okay, so it's just a theory.
No, he also investigated some, I think he cut up some dead brains.
Yeah.
And he found this. But of course, it was only the reason I say theory is because, he only created a hypothesis based on few evidence. But still, today, we don't have enough evidence to say that it always happened or that it sometimes happened. We actually have so few evidence on this that we have no idea, because we have not investigated it. We have not tested his hypothesis based, we haven't tested it enough. So that's why and the reason why I looked at this was because I looked at the images of these brain tumor patients, and brain tumor, we have not managed to prolong the living time for 80 years. Of the most glioblastoma, the most severe type. And then I was thinking, okay, if we have not managed to actually do something in 80 years, maybe we're doing something wrong, like completely wrong. And so that's why when I looked at the images, I could see that the place where the tumor was, then I could also see with my eyes that, okay, the brain actually has a very different metabolic pattern far away. I can really see, okay, if the tumor is here, I can look that the whole brain here has decreased activity. I can also see it in the little brain. And then, of course, started digging back on PubMed. Then I found Diaschisis. So, of course, this is also the beauty of science, that you see something, you get the theory, and then either you create the method yourself, or you go back in history and you see what other people have done. And then maybe they have done something you haven't thought of, but that connects your observation with an even better theory.
So you're inspired by a theory that came out 100 years ago?
Well, I was inspired by my visual sight. And then I saw that, oh, these great neuropathologists have actually produced science about it, you know. So that made me, that confirms that the path you're on, it's maybe something there.
You were telling me about two competing Nobel Prize winners from 100 years ago. So is one of them theorized Diaschisis?
No.
No, okay.
I actually just discovered this at the end of my PhD thesis.
Yeah.
But that it made very much sense. So after then I always talk about it, like I always knew it, but I haven't.
Tell me about it again.
The point is that in 1906 they gave the Nobel Prize in Medicine and Physiology to 2 people. And then you can show the image.
Okay.
And of course, here's the image of 3 people. And so one of them, so you can of course guess that it's only 2 of them that get the prize. One of them who was competing was Camillo Golgi. He is known for the reticular theory, which states that the whole brain functions in one network. Reticular means network in Latin. And so he stated that, and he stated that from looking in the microscope in his lab, okay? Then you have the Cayal, which was from Spain, and he said that no, it's completely wrong. He said that the brain only works in independent networks, so it doesn't work in a biological unit. They're not biological connected whatsoever. And then you had Fridtjof Nansen, which is Norwegian. I'm very proud that he's Norwegian.
Yeah.
And he said that, well, I think I have discovered something that connects the neurons, but it's not the anatomical unit. It's just a seat where I can see some black dots. And that's later known as the synapse. And of course, only 2 of these got the Nobel Prize. And it was not my fellow Norwegian, Nansen, but it was Cayal and Golgi. And they were so angry on one another that they had to give the Nobel Prize on 2 different days, because they could be violent too.
They wanted to kill each other. The brain's connected. No, it's not. No, yes, it is. Okay. Well, that sounds pretty aggressive now. But I'm thinking about it. You know, this Cayal, you know, the brain is totally separate out into different sections. That doesn't really feel inherently. It makes inherent sense to me. Does that make sense to you guys? It does make sense to you that there's different parts of the brain that aren't speaking to each other.
Yeah.
Okay. But yeah, there's been some people who've got the connection between the left and the right hemisphere severed, and it has, you know, drastic implications. You know, they can no longer, say, control one of their hands. You know, so that alone it suggests that the brain is certainly connected.
Yeah. Yeah, it does.
Yeah. Well, do you know
if the brain is connected?
Did your work support either theory, that the brain is
Well, our work supported the theory of actually all these 3. You know, because of course we do know, because the thing is that in 1906, the problem or the thing was that Golgi, he was politically abandoned, like he was put out in the cold. So people said to him, you know, even though he won the Nobel Prize, in general people said, well, he has misunderstood something vital. they said Golgi is saying that all the neurons are actually anatomically connected, right? But Golgi didn't say that. I read his Nobel speech from Stockholm, and he didn't say that. But the political environment or scientific said that he's wrong. So ever since we have followed Kayal's theory very much. So by evidence, by science, we do know, like you said, that yes, there are of course stronger connections between some parts, or maybe only connections between some parts. There are some parts of the brain that produce some things that is associated with anxiety or memory, or So we have explored his theory very much. And of course...
And so his theory is where the medicine textbooks are
yes,
based.
it's based on.
Yeah.
And there you and then you can see, okay, so you have the science saying, the thing is that, the medicines we take today, for example, working on serotonin levels or dopamine or these things, they are of course based on the neural doctrine, that if something happens to the serotonin receptors, well, you have to do something in that area, and then the brain will be better, right? So that's also why, very much of how we perceive treating the brain is based on the neural doctrine. And then secondly, you also have MR images and CT, which is what you do today, normally. And that is, MR image is also just a structural image of the brain. It shows how big your frontal lobes are and the gyruses and these things. So then if you have something happen here, you can also sort of try to theorize where that connection will go and where it will damage. So in general, we have done a lot of science and also science and treatment based on neural doctrine. But the other Nobel Prize, the reticular theory, we have not done so much science on. And of course, today we have a very big challenge in brain science and in treatment of the brain. There are very many diseases we don't know what is causing them. Schizophrenia or dementia. There is no, two lines under the answer. If it was, I would not be so enthusiastic about my own science. But yeah, because we don't have the answer to very many of the brain diseases, the most likely answer to that is there's something we don't understand.
Yes, 100%. But before I ask you specifically about your PhD discoveries, are there any visual differences between brain cells in various regions of the brain?
If there's any
visual differences, so if you look at a brain out of a skull, it all looks homogeneous. It all looks like the same, But if you look at our organs, they're very visibly different. If you take the front of the brain off, look at it under a microscope, or if you take the back side of the brain and look at it under a microscope, are there visual differences in how the cells are structured?
I'm sure it is, but I don't know much about it.
Okay. Yeah.
So I think I cannot give you a good answer on that, because and this is of course how.
So the question is, the question I have is why is the hypothalamus responsible for these things whenever
other parts are not?
Exactly.
Yeah.
Can we see visible differences?
I think, first of all, I'm not the expert on the small parts.
Yeah.
I am what I'm good at is that I know about diaschisis, and I'm good at technology, neuro software interfaces, how to use statistics in order to create a test. So in that sense, it would not be reasonable that I answer you because I'm not the expert. But
well, I'll get a brain. I'll get a brain, and we can dissect it over a couple of beers.
Yeah. But I think in general that, for example, lately it's more and more science produced on that memory. It's always been a scientific truth that it's been connected to hippocampus and these areas, right? But now lately it's more science produced that the whole brain is actually, there is, for example, one study showing that there's a network in this hemisphere of many different neurons, and they could see that in subjects that had less memory, this network functioned worse. So of course, this is also a question of technology. And I also think this is one of the plausible answers to why Golgi, why people sort of made fun of him. It was because they did not even have the technology to investigate his hypothesis.
It wasn't because of his mustache.
Are you trying to say something?
Your own PhD then,
Yeah.
Ivan. What discoveries came out of it? It was, I guess it was. It took 3 years to
Yeah.
to work on the PhD.
Well, it took 6 years.
6 years. Okay.
Okay, I started in a medicine study, and then I did, and then I used 1.5 year to finish it.
Yeah.
After a study. And well, we published, we published that the method I made by programming in neurosoftware using the correct statistical method, we showed that in brain tumor patients, when you measured the whole brain as one biological unit, we could predict if the patient would live short or long, so above or below a year, by looking at the whole brain and not only the tumor. And then we used the same method applied in dementia patients, where we also could show that the more the whole brain had a lower metabolism of sugar, and all the neurons use sugar, the higher was the likelihood of having the diagnosis of Alzheimer's disease.
So you mentioned Alzheimer's in connection with dementia.
Yeah.
Are they related?
What do you mean?
Because you started off, you looked at patients with dementia.
Yeah.
And then you used a model to predict whether they would get Alzheimer's.
No, not a model to predict, but of these dementia patients we had, some of them had Alzheimer's disease, and then some of them had a diagnosis of mild cognitive impairment. So, and then some were healthy controls. And then when we used the method, we could see that the healthy controls had a metabolism that was normal, defined by some parameters, and that Alzheimer and mild cognitive impairment had the metabolism that was lower in the whole brain.
Is that Alzheimer's? Is Alzheimer's a lower metabolism in the brain?
It could be.
Because we don't know what it is at
We
the moment,
don't.
do we?
No.
Do we know what dementia is?
Well, I maybe get hanged if I say this, but I would say no. If we knew what dementia was, why don't we have a treatment?
Yeah.
Or why don't we, are close to a treatment? And of course, as far as I understood, this can be wrong, but we have not it's not been published a new medicine on Alzheimer's for the last 10 years or something. And so if you ask me personally, because again, I'm just expert on technology of software and diaschisis, but personally, of course we don't have an answer to dementia. And this also goes to, so one part of the thesis was about looking at the brain disease that spreads in the whole brain, but the other part of the PhD was to create a method that is technologically good enough. Because there are so many different softwares that is produced commercially by companies, and there are so many different methods. But in general, very many of these softwares that these companies sell, they have these black boxes of statistical working. So they're producing numbers, but when you really look at it, it does not connect back to the biological source.
Well,
so it creates some biases.
You're going to have to explain to me exactly how you go from software on a computer to, sorry, just turn that on silent there, my phone's going off. How do you use software and computer programming to look at the brain?
Well, it's like, we talked, it's the PET images that I looked at. So it means that
Is that with the nuclear,
Yeah.
the nuclear medicine? Okay. Everybody remembers this? Yeah. Sorry.
So you
Got another phone call there. Yes, go ahead. Skål, everybody. Are people allowed to ask questions on the way?
Yeah, all the time.
Yep. Go ahead.
Just ask. And tell me if I talk too much, because I could talk very long
Well,
phrase.
I want to hear about it. So come on. Software, computer,
Yeah.
nuclear medicine.
Yeah.
Tell us
Okay.
the
Well,
connection.
the thing is that you inject reactivity into the blood veins.
Yeah.
Okay. Then the patient is resting for about 45 minutes after injection, so that all the reactivity can go out in the body. And then you put him in the scanner, and then when he's lying there with his head, the reactivity, of course, is beaming out in many different directions. And then you have cameras 360 degrees around that they catch these reactivity rays, and then they go to the computer, and then the software looks at the coordinates, and then the coordinates creates the image. And then you can put onto that image of raw coordinates, you can put the software on. So if you have a brain tumor, the tumor will eat much more sugar. And the sugar is connected to reactivity that we have injected. So it means that we look at the image, the place where you have the tumor will light much more up, like a sun. Then other parts of the brain will look like a cloudy day. And that's how you can visually look, saying that, okay, the tumor eats more sugar, it has a higher metabolism. So in the first 30 years of PET science, because PET was developed in Philadelphia at the same time as MR and CT, which is structural imaging. But CT and MR, they're selling this much of these scanners today, and PET is here. It's because the technology is so advanced. And I don't remember my point.
It's a software.
Yeah.
There's software to get these images from the cameras turned into to areas in the brain that's consuming more sugar, and then you're coming in with a different kind of software.
Well, no, I use the software.
Yeah. Okay.
A very low-key software that I could draw the areas myself, and I could mark the areas myself in the brain. And of course, I was very lucky. So one of my mentors, after I had written my protocol, I didn't understand any of it, not the Ph.D., but just the first science protocol when I started. And then I delivered it, and then he came here. He was from Philadelphia. He came to a conference here, and then he said, somebody presented the data I was working on, the images, and then he said, "This is beautiful data. Who's working with this?" And I said, "Well, I'm working with it." And I said, "You should come to Philadelphia and visit me." And I said, "Yeah, I would love to." And then I said, "Well, I have no money." And then my boss here in Odense said, "Well, we pay for your plane ticket and everything." So I went there just maybe a month after I started my project. And the reason why I'm saying this is because he was in the group that did this first of everyone. He's like a pioneer. So he's been also creating the software that I was using. So of course in science, it is extremely important if you're new and you don't know anything, that you're actually connected to the smartest brains. But my mentor, he had always been, the ugly duckling, because MR and CT has always been the most popular, you can say. And when you have a biological image of the brain, it doesn't look very pretty. It looks like a Picasso painting. And also, when my mentor, Abbas Alavi, he's from Iran, he said when I was young working with these images, I could show the PET images to the MR doctors, and then they would just say, "Abbas, why do you do this? Go get another job. It just looks like a painting." And so meeting these visionaries so early in my science career, it really inspired me to believe in what I intuitively thought was right. Because logically, of course you just read what the textbooks say and you accept it. But intuitively, I think that's what science is about. You have intuition, you want to follow it, and then you have a methods to see if it's true or not.
And you followed the method, and you found some evidence to support your
Well,
gut feeling.
Yeah, but my problem was that it did not exist any method for looking at how diaschisis spread in the whole brain. And my mentor, he told me, when I said to him, "I read about this diaschisis, what do you think about it?" Oh, it's so interesting. I've made a publication 20 years ago and nobody listens to me. They're idiots. So that made me believe that, this very wise man, he also thinks there's something to it. And then I started using the software he had produced, but that was, of course, for just nuclear medicine in general. And I realized there are no methods. So then I started to exploring, is it possible to use this software to get a quantitative number out so that I can have a method so I can only push one button to get an outread of the whole biology. So it started with being interested in the brain, but I ended up with actually spending all my time in just trying to create this method in the software, use the correct statistics and all these things that I had no clue about. So I had to learn everything from the beginning. And that was really fun and really interesting.
That's good to hear. You know, it was really difficult, but it was fun.
Yeah, it was really fun.
I
You know, I think if it's easy, it's not so, at least for me, I find it more boring. But when it's complex, and of course this is the beauty with science, of course, you know that, okay, it's hard, it's stimulating for myself, but you can also smell that, okay, if there is something to this, you can really help many other people as well. So it's both individual benefit, but also a common benefit.
So tell us about the development of the method, because we're talking about the origin of the company, Synaptic.
Yeah.
So explain how the method works into you, along with some collaborators.
Yeah.
Starting the company.
Well, so I worked with the method, I developed it, and then we made a very nice publication. Then I went to a conference in Vancouver, Canada. I was standing in the line. It was a brain conference, and then it's like this guy in front of me, and like, should I talk to him? Okay, I talked to him. And I asked, so where are you from? And he's like, well, I'm from Copenhagen. And he's like, where are you from? Well, I'm from Odense. Yeah. And then what are you working with? I'm working with looking at the whole brain, metabolism. What are you working with? Well, I'm working with the same. And so the rest, I mean, he was almost the only person I talked to on that conference. Because I wanted to learn about his method, and he asked me questions about mine, and he made a method looking at the metabolism with sugar, with nuclear medicine, with patients with coma. And if you could predict by creating a quantitative method the degree of consciousness by using coma scales, in order to see how awake the person was.
Okay, wow. So if you're like way gone in a deep coma, you have low metabolism in the brain, I guess.
He was looking at this, and he had made some publications stating that yes, there was a theory about this. He had found some few evidence, and this was at the time that Michael Schumacher had this accident in the Alps.
Yeah, and he was in a coma.
Yeah, so he told me that, all these people would call him, these journalists, and said, well, you have this method, right? But of course, it's science, and he only looked at a few people. So he also made some great publications, but again, in few people, and his method was beautiful. So when I came back here, I additionally thought that, this is really interesting. So I got a hold of a new software that was an upgrade of the old software, where I could program myself much easier, like drag and drop and these things. So then I tried to mix his method with mine, and was playing around with the images of the brain tumor patients, but also with the Alzheimer. So I merged them, and I said, I want to make a method that detects diaschisis across the primary brain disease. Just like let's say if you had an idea, you wanted to create a method to measure blood pressure. That's also, you can measure blood pressure regardless of the primary brain disease. Well, why couldn't you do that with the brain? Mm-hmm. And then playing around, then suddenly one day I'm like, shit, I really think this works. And then like, nobody knows about it, and it's new. I wonder if I can take a patent on it. It was really cool. It's really cool.
Yeah. So again, I'm finding trouble to picture this here. So you're looking at different regions of the brain with different degrees of sugar metabolism.
Yes.
And you're seeing on this side of the brain, there's the damage there. So that has reduced or increased because there's a tumor there. And then on the other side of the brain, there's something related.
Yeah.
Okay.
And we can also look at the picture. I think we have one of
do
the
we have a
brain.
picture? Where is it? Is this it?
Yeah.
Okay. For the people on the podcast, we're looking at a picture. It's called the synaptic brain. On the left, what do we have here? We have a very colorful image with some red on the right and a little bit of red on the left, and the rest is yellow. And on the far right image, it's just all black. So, what's going on with the image over there on the right? There, I'm in the all black image.
Well, you can see there's a MR picture,
Yeah.
and so you can see that it looks more beautiful, right? You can see some of the details better of the brain.
Yeah, it's all black, but on the right there's a white spot. Is that a tumor?
Yeah, that's correct. What the blue arrow is.
Yeah.
Right? And then an MR shows structure. So you can see here that the structure show us that where the white area is, the structure is abnormal. Of course, because of the tumor.
Mm-hmm.
And that's what we use today if you have a brain tumor. That's the gold standard if you have it. And then to the left in the image, you can see the PET image of the same brain and the same patient. And now I told you about how tumors eat more sugar and these things. So maybe you can tell me where the tumor is.
Okay, so there's three different colors. There's green, there's yellow, and there's red. And I say the parts with red is the sugar consumption. So where we know the tumor is, there's a lot of red, and then on the opposite side of the brain, there's a little bit of red. So yes, so it seems to be almost not fully symmetrical, but there seems to be some kind of symmetry going on.
And where do you think the tumor is?
I think the tumor is over on the
this one. Oh, sorry, they are.
Oh, they're mirrored. Okay.
De är spegelvänt.
Okay.
So this here is actually the others.
So where the tumor is on the MRI scan,
it's on a different side.
There seems to be more damage to the brain on the opposite side of the actual tumor.
On the MR image.
The tumor is here.
Yeah, that's correct.
But there seems to be much more widespread damage on the opposite side of the brain compared to where the tumor is.
Because it's red.
Exactly.
Okay, half of it was correct.
I'm not a brain scientist.
Yeah. But it's cool. But you, so of course, so you are right that the tumor is here because it eats more sugar, so it's more red.
Mm-hmm.
But in general, we say that okay, this is the healthy part because it also have some sort of sugar metabolism. Because then if you think that okay, you have diaschisis, meaning that neurons are connected all the way in the brain, but also when you have the little brain here, there are much more connections going across than which is going here. So you can see that the little brain here is very much affected metabolically wise compared to the other cerebellum. And you can also see that the whole hemisphere here, on the same side, has also decreased metabolism. And today, we only look at the tumor. You know, we remove it. There's, you know, highly specialist neurosurgeons but we don't look at these parts. And this could be one of the reasons why we not have come a longer way in treatment of brain tumor.
Well, whenever you remove a tumor, do you see a difference in the rest of the brain? Does the rest of the brain know that the tumor is removed? Because if your theory is true and the rest of the brain is affected by the tumor, if you take the tumor away, whatever is connecting the different parts of the brain, and I don't know that yet, that mechanism is removed as well. So after the tumor is removed, have you done the same scan?
Yeah, we have done. We follow them for 3 years.
Okay.
And we just saw that in general, also after the tumor is removed, if your brain still have a low metabolism, it is associated with a short time to death.
Yeah.
And that's the only answer I can give you. In general, nobody knows the biological mechanisms of diaschisis in humans. It's like the science has we're not come very long.
So your work has just proved that the Diaschisis is something that we need to consider, but there's no mechanism discovered yet.
Well, the PhD has just shown that we have created a method.
Yeah.
So here's the method. Now the next step is to apply it in a larger group of patients, because even though we are published in good journals, we have no clue if Diaschisis is so relevant as we think before we have validated it in a bigger group. And then the last work of the PhD was a systematic review, where we looked at all the science that has been done on Diaschisis. So it's roughly 3,000 patients that have been investigated by different scientists, but they all have used different methods. And of course, if you want to look at a phenomena in a larger group, you have to use the same method. You cannot use 15 different methods. But we saw that Diaschisis had been found in very many different parts of the brain, in different connections, different independent connections. And thus we created a synaptic map, a network Diaschisis map. And also the test that we made, we called the Network Diaschisis test. And it was not I had one of the best brain scientists also as a mentor, who said that, oh, we should name this something new. So from this, the end point of the thesis was that it was the naming of a new brain disease, network Diaschisis, meaning that it's not only Diaschisis in one neural connection, but it's actually Diaschisis in the whole network, in the whole reticulum. And to end up your first question, so the work on the patent was to work on combining mind Diaschisis method with the consciousness method from Copenhagen. And then I added a brilliant mathematical scientist and doctor, which is, we've been partners for 5 years. And then the result was that we gained the patent on the whole brain in a year ago.
So you made this discovery of connections within the brain because you're the kind of person that goes out and you make connections yourself. You talk to people. It sounds that way. You're just curiosity driven. You're just talking to people, trying to find out what's going on.
Yeah.
Bumping to people in a queue in Vancouver, discovering ideas and bringing everything together. I find that fascinating, and that's what Science and Beers is about. You drink beers, you talk to scientists. There's one more thing I want to talk about as well. It's the next thing that this group of people that you just mentioned went on to make. I believe it's the COVID tracing app.
The COVID
tracing app.
Yes.
The AI behind that.
Yes.
And how that connects to the brain.
Yeah.
Can we take a break and have a beer first, though? Is that okay? Sure. Yeah. Thank you very much. We'll be back in 10 minutes, will we? Okay. Thanks. This works, yeah? Oh, you have to turn that on, yeah. Turn yourself on. There we go.
There.
Can we close the door there, please? Thank you very much. So we just learned about Ivan's motivations, curiosity, brain disease, diachisis, and we're going to go on to talk about how this relates to COVID-19. But first, we're going to watch a little video, okay? Here we go.
One of the most efficient means to fight the pandemic is to keep infected people separated from healthy people. Identifying carriers is therefore critical, which puts a lot of pressure on our hospitals. People are currently being tested either individually or in smaller groups of 3. There's a big potential in identifying the carriers through testing of bigger groups, and our scientist Sophia can determine the mathematical optimal group size for each individual. If the group test is negative, then the whole group is clear. If positive, then every person is tested individually. This demands less analysis, we get the answers way quicker, we reduce hospital expenses, and actually the test will be more accurate. Our solution is to combine group testing with a unique worldwide data sharing system for improved analysis. The patients are first led through questions on their phone, where the Dr. Jacob is investigating the patient's symptoms and signs. The data is then analyzed by Sophia, and patients are being put in groups based on the likeliness of having the virus. We have created a synaptic system that makes the handling of sensitive data way easier because personal data never leaves the phone. This solution will boost the economy, leads to less quarantine time and reopening of schools and education. This means people can now travel and attend social events like concerts and football matches.
A Synaptic Science Solution fitted for a global network that makes big data small.
Does that not sound interesting to people? So, I for one would like to some kind of mass testing, which would mean that I could, which would mean that Bruce Springsteen could go back on tour again. So Ivan, how did this come about? First of all, what is it we just saw? We just saw like an advertisement for an app that's based on artificial intelligence.
Yes.
That can mass test.
Yes.
For COVID-19.
Yeah. That's what you saw.
That's what I saw. How did this idea come about?
Well, it was because, like we ended the talk, my partner, doctor and mathematician, scientist, we made the brain patent, and then now when the COVID-19 came, I asked him, well, we should do something. I think this virus, we should try to do something. And then I didn't hear anything from him, and then maybe a week later or something, he returned and he said, I think I have something that could work. And what could work was actually the reason why we met. We met at the conference in 2015 where I presented what I have done with the brain, and he presented a mathematical equation he had made for chlamydia testing.
A mathematical equation for chlamydia testing?
Yeah.
Yes, okay.
And this he had then translated into use for COVID-19 testing, but additionally he had the program in open source software. So he was not finished programming, but he contacted me and then he said that, if we do this and this, we can group test, hence mass test, large number of people for the same cost as a small number of people. And then not very long after that, the group from Israel published a study confirming his theories, saying that we have tried to group tests of COVID-19 in a laboratory, and we have seen that you can group up to 30 tests of COVID-19. So the saliva from 30 people into one pool, then you use one COVID-19 test into that pool, and if it's negative, it means that there's no virus in that test.
You're talking about the standard test that is PCR?
Yeah, PCR,
yeah.
exactly. So it means that if you do that on 30 people, or 32, you have actually saved 31 tests. And we all know that the COVID-19 costs money. So it means that that's group testing, and that was developed during Second World War by a professor called Robert Dorfman in test, because the syphilis epidemic, and they had just produced and created penicillin, but it was not enough penicillin. Hence, he used group testing to figure out who to give penicillin and who not to give penicillin. So it's an old, again, it's like Diaschisis. It's an old scientific principle.
So he had a load of soldiers and put all of their, what was it, blood
Yeah.
into a jar, tested all of the blood for syphilis.
Yeah.
And then
Then if it was
Might be,
negative,
yeah.
it means that you don't have it. But if it's positive, then you retest the group. Then you test them individually, okay? So it means that this group testing, you do not lower the analytical accuracy whatsoever. You just make a more efficient system.
It makes total sense.
Yeah,
Get as many people as you can. If people want to go to a football match, they all get
exactly. And
tested at the same time.
it goes faster, because if you can, a PCR machine, often the standard is that you have 96 places, 96 tests, and it shakes it for 4 hours or 6 hours, and you get the result. But instead of having one test in one glass, you can have 3 tests or 5 tests. So that
Or
lowers
30 tests.
or 40 tests.
40 tests in one vial were, okay, and then you have 96 of them.
Exactly. So you can see how exponentially you can really start talking about, let's say, testing 80,000 people, because it's in science has there has been science published showing that you can save between 30 to 80% of all COVID-19 tests if you use group testing strategy.
But PCR, it's polymerase chain reaction. It amplifies a certain part of the RNA in the virus, and if I was getting tested, it would be a swab from the back of my throat, which would end up in a little vial like that. But if there was 32 people going into the same size of vial, the worry that I would have would be that that little scraping from the throat or the saliva would be so diluted that any one individual who did have it, and they might have a very low amount of virus in their body, it wouldn't show up in the PCR.
Yeah. So of course that's a danger. Hence, if you start doing group testing, they do it here in Odense actually. They are grouping 3 people. So every three people are grouped in one test. So meaning that if you want to do it on 30 people, the study I referred to was just a exploratory study.
Yeah.
So it means that this is a field of science, just like Diaschisis, that we have not put much attention to. So of course, if you want to really figure out how many can we group, then you have to both test individually, and at the same time you put the same test, you divide it, and you put it in groups. And then you see how accurate can we be. And then let's say if you test it on 10,000, that okay, we can actually have 4 in a group. Then you continue to 5 and 6. And then of course, if you start doing this, then of course the industry will also start exploring what chemicals can we make so that we can actually make group testing more efficient. So it's not like you can just start testing 30 people in a group. There, of course, it's steps.
Yeah.
Yeah.
But that's I know that this group testing is an idea that's coming in. I've heard about it in the news in the UK, Is it happening in Denmark? As people, well, we just said they're measuring in bulks of 3. But then how does the AI come into this? How does your...
Yeah, well, the thing is that Sophia, because the thing is that you know,
Sophia is the name of the AI, yeah.
yeah, if you just group test randomly, if you take all us in this room and you said, okay, group of 3, group of 3, group of 3, it economically it makes more sense, and also time efficiency, that if you have been traveling, but all of us has been here in Denmark, we test you alone, right? And then because the likelihood of you having COVID-19 is a little bit higher than all the rest of us. So the AI comes into play because it says that these people have a higher likelihood of having COVID-19. Hence, when you actually perform the group testing, those who have a likelihood is tested by themselves. And those who have a very low likelihood is tested in groups. So it means that you higher the statistical probability to lower the number of groups you have to retest. And that in itself can save enormous amount of money and time. And the way it works is that, well, you download the software Jacob to your phone, which you have produced, and then you put into variables like fever, yes, no, cough, yes, no, many of the same things that you actually do on Test Denmark's site when you go in there to get a test. So then when you, and these variables we have gathered from scientific literature all around the world of characteristics of COVID-19 patients. And then when you have entered that, it takes only 1 minute, you push analyze, and then the machine learning algorithm, which is Sophia, she tells you, okay, you should be grouped by yourself, or in a group of 3, or in a group of 5. And this number you can then give to the test person, so they can put it on your test, so that the laboratory knows if you'll be tested by yourself, in a group of 3 or 5, and so forth. And
okay, so you're limiting the amount to 5.
No,
not 30.
The AI is created to larger groups of people. The reason I say 5 is that, for example, now we're testing group of 3, but maybe 5 we could do today, but higher than that we need to do much more science in order to be sure.
Okay, yeah.
Yeah, and the other point with the AI is that you know, if you, let's say if you do this study in Canada, you get valuables into the machine learning algorithm, and if it gets smarter, then it will, Sophia will leave the phone, but it will not take the sensitive data with it. So when you use the algorithm, it will never take information from your phone. And that makes that you're more safe, we don't need any cloud, and it also makes Sophia smarter. So that's also what we find, what we think is important. Because today, everything, you know, the data we have is often exploited.
Indeed, but there's this thought in the consumer maybe that who can we trust? Like there's many people say we'll not take your data, but will they? Is that true? You know, but how does your idea for this app compare to, say, in Denmark, Smittestop? How many people have Smittestop? Half people in the room have the Danish app Smittestop. How does Sophia compare to that?
Well, it compares because Smittestop, and again I'm shooting from my hips, but it's not a science app. It's a surveillance app. It does not create anything new. It just it's just a system to figure out where you are. But our app, it actually, you know, because it's a machine learning algorithm, an AI, the more data you put into it, you can also, every time you use it, it marks what location you have. It means also that it gets better in pooling correctly into groups. So, and the reason why it's important is because you have very many different machines performing tests in the laboratory. You have Roche, you have Siemens, blah, 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 blah. So it means that if you want to enhance science on group testing, if you have a system that can, you know, Sophia, that they can actually travel around all the places, it is easier to systematically understand how many can we pool. So the difference is that, and boldly we say that, well, we really think we have created maybe the first science app that actually you can connect everyone who wants to use it.
Would your app, like Smitastop, for example, the way that works, it uses Bluetooth, and it can say if anybody that you've been in close proximity with over the past 14 days have tested positive and have typed that into the app, it will just give you a notification to say that, and to say that you should go into quarantine for the next 14 days. But there's a lot of problems with that. You know, if I was in contact with somebody who tested positive 14 days ago, I don't need to go into quarantine for 2 weeks, because the chances of me picking up from them are quite limited, because they've already done their 14 days or their 10 days quarantine since they were infected. Likewise, if I was in contact with somebody on the day that they were just infected, that wouldn't be able to tell me that, you know. So does your idea also inform people if they were in contact with somebody else who tested positive?
Not yet. No. And this is
But your app is not to let people know that they were in contact with people who were infected. Your app is to test as many people as possible in
as fast as possible.
As fast as possible.
And that's why I also say our app is a science app, because it tells you yes or no if you have it, and the theory.
And who you can go and party with.
Yeah, but in theory, it is a system that if you want to have a music, a concert, if you used, and let's say you have 10,000, you can actually test everyone in theory. You can, because the problem with testing is that we have not enough COVID-19 tests, or we don't have enough money. It costs too much. If you want to pay a private company to test you at the airport, it costs between 1,000 and 2,000 kroner.
Yeah.
Right?
Yeah.
So imagine if that company had a group testing strategy. It will lower the cost between 30 to 90% for each test. So
the whole I traveled 2 weeks ago, and I also wanted to get a test, but I'm thinking, fucking hell, 2,000-3,000 kroners for a test. If the whole plane landed and the whole plane was able to all spit in a bucket and then see if anybody was infected, that would be a very useful tool.
Yeah.
A very useful tool indeed. Yeah.
Yeah. Or let's say there's so many things to this test strategy that I think doesn't make sense. Like you say, okay, you take a test, but then you actually meet other people, and then you get an answer.
Yeah.
Well, then you actually meet many different people.
Oh, yeah. Indeed. Yeah.
But the thing is, and I think the challenge is that it is economy, and it is that we don't have enough equipment. So we have to create a method that use the resources better, and you have to create a method. And then this is the beauty of science. Good science, it tells you yes or no. You don't take a guess. It is yes or no. So instead of guessing, okay, we take a test, how many people do I meet? I use Smitastop app. I figure out maybe this, maybe that. It's not science. Science is yes or no. Hence, our aim is to make a tool that is political independent. Because right now we are, the politicians are, have power of all our lives, and they're doing their best. And the problem is that their tool is lockdown.
Yeah.
But the lockdown creates many people are losing their jobs, many people are isolated, and these things. So if you have a tool where you can get people more out of lockdown, that is political independent.
Yeah, and it's completely.
You see what I mean?
It's crippling the economy as well.
It is crippling economy,
Yeah.
and the people on top, of course, they just get richer, but the people in middle and lower, they really suffer the consequences, not only now, but in many, many years to come. So of course, this has also been our intention to create, I mean, we have programmed the whole AI without any money, without any investors. My partner who has programmed it in open source software, then we have connected him to another programmer in a different country, which have created the software itself. You know, we have come so far because, and we work, you know, in our spare time in these things, because we really think that the testing regime that we have today, it is not good for us. We need something better. And it is possible. It's really possible to create something where we don't have to suffer for 1 or 2 years. Because the longer we suffer, the more exponential is what happens after, sort of.
How do you have any spare time?
What?
You sound like a very busy man. You
Yeah.
know, so you're studying medicine, and you're doing your PhD, and then you were part of a team that created a COVID app.
Yeah.
You know, that sounds like your days are pretty busy.
Yeah, they are.
Yeah.
It's annoying. It really is annoying.
Yeah.
Yeah. So.
Also, Sophia and Jakob is the name of your kids as well.
Yeah, that's the name of my daughter and my son.
Okay.
Yeah.
So a family man as well. So
Yeah.
so very busy. But this is for the good of humanity, is to get the economy back together, is to get mass people tested. And from talking to you, this is completely new territory to you.
Yeah, for me.
Yeah,
Yeah.
you've not done anything like this before. You haven't invented an app or
No.
or an
No.
AI before. So I guess you're learning as you're going, but you've came so far. You've developed the app. You've given proof of concept. How does one go about getting this app out into the world?
Yeah. Well, you tell me.
Okay, yeah, you're still
You tell me.
you're trying to figure it out.
Yeah.
Yeah.
Well, you know, it's my partner who has created the algorithm, who's programmed it. Then, of course, there's another person who has created the software, and then we're all working together on trying to expand this, and that is challenging. It's really challenging. You know, you have to have a proof of concept, or you have to have investors. But you know, it's a very unholistic world we live in today. So, and our solution is very holistic. So we also work towards, you know, we made the app, but then working with some laboratories, we understood that, okay, it actually does not exist a robot to perform the group testing. It exists some, but they're not very efficient, and they cost many millions. So we started looking at a robot, and we realized that, okay, it will only cost like 40, 50,000 to create one. Maybe we actually could create it like an IKEA robot, blah, blah, blah.
Why can't you use a standard PCR?
We can,
Test
of course you
robot.
can. But the thing is, I mean, there's many things to it, but I mean, one of the reasons why everything is so challenging right now is that you have the healthcare, the hospitals, you have the scientists, you have the commercial companies, you have the politicians, but they all have different interests. So the robots that are in the laboratory, maybe they cost a lot of money, but the scientists have no other option than to buy what is offered from this American company, let's say. But in reality, they maybe need a much smaller robot that works faster and is cheaper, right? So when we started looking into this, we also work with trying to see, is it possible to, we know it's possible, we've been to meetings, it's extremely easy to make this robot. It's extremely easy. And most likely we can actually make a robot, all the parts exist, where you actually can, in theory, just order it by email. And the reason why this is important to us is because we're creating a solution that we want them to be possible to use in refugee camps and in India and countries where we don't have all this money that we have in the West. So how to come from that, we have this product, we have created an AI to actually get it out there. We have worked very much with it, but that's the challenge right now. And of course, you know, our vision with the company, with Synaptic, which was founded in 2018, is that the brain AI that we now have a patent on, and then the COVID-19 AI, it can actually be used in many different diseases as well. We want to combine these two, because then if you combine it, it's a way of reporting symptoms and these things in a new way. Hence, of course, we would like to get investors in, but the problem is that, you know, we want science to be the main force, what leads this project. Because we know that, you know, of course it's good to make money, but it's better to make good science if you really want to help someone. So these things are complex, it's challenging, yeah.
But so you're looking for net investors, and not being a politician myself, you know, this is a money saving thing for a state, you know, if they can mass test, you know, so spending a few thousand kroners for a PCR machine doesn't seem like a lot in the greater scheme of things. Whenever you take out the price of paying the individual scientists for doing the individual tests for individual people, you know, so it sounds like something that a government might be interested in, not to put money in, but to say, okay, we can save this amount of money, X amount of money, if we take this on board. Have you tried to approach the government from a not giving us money, but them saving money perspective?
Do you want the honest answer or the diplomatic answer?
A total lie. Give me a total.
A total lie.
Of course, the truth.
Yeah, they're so
truth.
interested.
Give me the truth.
Yeah, they're so interested, and like, you know, they're very welcome, and you know, everything.
So you've tried this approach to it.
Yeah, well, of course, I'm from Norway, but I live in Denmark. We have contacted so many of the Norwegian authorities, but also Danish authorities, and it's so far it has been impossible to come through. Just impossible. You know, we send them the pitch, blah blah blah, but you know, it's all in the name. It's all who you are. Everything is network today.
Well, that's how you came here in the first place. That's how
that's true.
That's how you got to the place where you're able to create the AI, because you talk to people, because you have a network, because you're not shy to talk to people.
Yeah.
You know, so you more than many people know how important network is, and I guess whenever you're trying to influence policymakers, it's probably a complicated process.
Yeah.
You know, and given the structure of the society that we live in, no one person is in charge. There's a certain way of doing things. It has to go through certain procedures. It has to go up the ladder, so to speak. You know, so that mustn't be easy to do without that name.
Well, it's much more complex than the science.
Yeah,
it is.
Yeah, so
but you can show the slide where you can see how you know, some of the estimation of what you can save.
This is the savings here, is it?
That's daily.
Change it over here. So, is this the slide that you're looking for?
Yeah. And this is estimation of. Right now, we're testing each person individually. But then if you compare those costs every day of that you can save between 30 to 80%, this is actually the number you can save each day. And the reason why this is important...
Each day?
Yeah.
So, sorry, how much is Denmark spending on COVID testing at the moment?
A lot.
Okay, wow.
I mean, the thing is that, you know, the reason why this is important is that, I don't know this for sure, I haven't checked it up as a fact, but I talked to a Ph.D. student at the hospital, a doctor at SDU, and he told me that, you know what, SDU now is actually holding back Ph.D. salaries because they don't have more money because of COVID-19 testing. He told me, I don't know, he told me that now, because of all the money we use to COVID-19 testing, you know, before, if you published a paper, you would always get it sponsored by the university. But now, it has actually become a selective process, meaning that there's some journals that you get sponsorship to publish in from SDU, but some you actually have to provide the funding yourself. So then you have to ask your professor, do you have money to publish here? So of course, this is why it is important for us that the government saves money, because all this money for testing is taken from a budget that of course very
Well,
often goes.
if I'm flying into an airport, I'm paying 2 or 3,000 kroners for my test, you know, and if the Danish government are testing like hundreds of thousands of people,
yeah,
that could add up. Yeah,
it's insane a lot of money.
Uh-huh.
And there, I think, you know, if you are to defend the politicians, well, their challenge is that they are not scientists. They don't know the details of the testing regime, meaning that they have to take decisions based on reports they read from others. So, you know, this is also, I think, why some decisions are maybe off the chart sometimes. Because, you know, we should have, scientists should be more, I think, involved in the decision-making process.
Mm-hmm.
And for example, a solution like this, I'm not saying that it will work. I'm just saying we should try it. And then please show me that it doesn't work.
Well, quite recently in Denmark, there was the mink crisis with the COVID,
Yeah.
the cluster 5 COVID mutation. And only this past couple of days, there's been some science to come out to say that actually the vaccine that's targeting the proteins on the surface of the vaccine, even in the mutation, the vaccine will still work. But they still took the decision
Yeah.
a week or two ago to kill every single mink in Denmark. So it is possible to like make these very spontaneous decisions in government to even.
And in that respect, you know, if you had a group testing regime, of course I'm biased because this is our baby. But you know, if you have a group testing regime, it's a tool where you can test very many people or animals very quickly. So instead of taking decisions out of a gut feeling of what you think is right, you take a decision out of a gut feeling of what you know is right. So this is also, you know, one of the core values of our company. It is that, you know, we really want science to prosper, because instead of going from systems where we think we act right, we follow a system where we know we're following the right.
Yeah. You probably need some kind of like test space, you know. For example, in Germany, a couple months or maybe a month ago, they set up this test where they invited a bunch of people into a concert at a large music venue, and they used those people as test subjects to see how COVID spread. You know, you probably need to start small and take a certain area or maybe an island, and see if you can approach the mayor or the local council there to see if you can introduce this app, to see if it works there, and then scale it up.
Yeah.
For example, approaching maybe the mayor of Odense and saying, "Hey, let's test everybody that wants to go to an OB football match."
Yep.
And then that it starts there, and then it gains some traction.
We are working with 2 Danish hospitals in order to try it out.
Okay.
And hopefully we can, you know, try it out at least one of these hospitals very soon. So it is, we are crossing our fingers that we can do that.
Yeah.
And so of course, because if you can do that, if you can show that it works one place, then you can implement it other places.
Yeah. As long as I'm able to go to Bruce Springsteen concerts again in a year or so, because he just came out with a new album there. Let me see. Yeah, that's your team there behind Synaptic.
Yeah.
So you guys you made a company, and what you're doing is you're inventing a way to test the connectivity of the brain and COVID.
Yeah, that's true.
Yeah.
And I think in essence what we do is that we just say we want to use the best science, the most logical science, and then we want to focus on that with the new technology in hand. So that's this, you know, both with the brain and the COVID. I mean, we're using scientific principles that is either 100 years or 50 years old. And we have programmed either algorithm in open source software or in the neuro software. It has been extremely cheap to make it. And of course, we do it because we think it's fun. And we want to try it in larger materials. So that's like, and then I think other part is that we live in different countries. My partner, who you see there, you know his name, you've seen his image, but he would prefer to be anonymous. And but he's a really nice guy, even though he has chosen that image. But he lives in a different part of the world. We have collaborated on WhatsApp digital platforms for 5 years. Thomas, that is here, he lives in Slovakia. He created the Jacob software. We all collaborated on digital platforms. It's extremely efficient. And then Vegard, that is here, he's a co-founder of a medical app that has been very prosperous in Norway and knows much about commercialization of these things. And so, you know, we don't even live in the same countries. So that's also part of the philosophy that, you know, we have all the, you know, it's like we have this brain today, which is the internet.
Yeah.
But still, I think there's so much chaos. And I think we need some sort of synaptic energy.
Yeah.
And
Diaschisis.
Diaschisis or an entry point where we think differently. Where maybe, you know, we think in a millennial way. And maybe not in the way the generation above us thought. I mean, that's the meaning of generations. We are to evolve and create new systems. But that also means that we have to be bold and we have to follow what we think is right, even though very many others say it's wrong. So this is also where we're coming from, that we really want to follow what we really think is right for our generation, but also for those who comes after us. And that maybe means to break some of the older system that is governing the way we think today.
I think you painted a picture quite well with the two Nobel Prize winners 100 years ago. That they had two different ideas of how the brain works, and they hated each other. They couldn't even be in the same room. Whereas today, you meet somebody with a different idea, and you're like, okay, let's talk, let's get a beer, let's see what we can do, let's brainstorm, let's build up on our idea. And that's how you got talking to your colleague in Copenhagen, for example, whenever you met in Vancouver. So I think your whole talk today, and it seems to be your whole philosophy, is about connectivity.
Yeah.
You know, so you meet people, you talk to people, you inspire yourself, you get ideas about the connectivity of the brain, about the connectivity of other people thinking about the brain, and then the connectivity about the app, how we can come together through the app and test for COVID so we can eventually make it to Bruce Springsteen concerts again. So I think that was a very inspiring talk. And I think one thing that lubricates connectivity is, of course, beers. So cheers.
Cheers.
Thanks very much for coming out today. Ivan, are you going to hang around for a couple more of these?
Yeah, I like connectivity.
Yeah. Thanks for coming, guys.
Thank you.